Here it is, Bikini. This is where it started. Before that, 1946, the peace and simple life of Bikini had changed very little in the course of countless generations. The only interruptions were storms, which sometimes drift into the marshes from the more turbulent areas of the Pacific. Bikini's beaches had been used for the outriggers of young men who sailed and fished in the lagoon. Coconut palms for food and clothing and shelter for the people who lived simply. Bikini Lagoon, the stage for an operation of men and ships and machines, and to a top Operation Crossroads. Everything that existed in this tropical circlet of coral became a part of the physical transformation and enormous human activity that produced facts we're after now. Operation Crossroads, the beginning of full-scale controlled tests of atomic energy. The beginning of peacetime research by the civilians formed to meet the specifications of the experiment. This was the means to get at the facts. And now all we need is time. Time to trace the intensity and effect of radioactivity through every type of life at Bikini. Now, Bikini is quiet again. But under the silence of the atoll's waters, something is still happening. Something that must be studied and measured. The lagoon still contains traces of energy released there on Baker Day. Radioactive energy. For us, the biologists, the story of Bikini merely began in 1946. For us, the far more significant story has been unfolding year after year since Operation Crossroads. The long-range problem of whether these traces of radioactivity have observable effects on various forms of life. The problem of how radioactive materials persist and circulate in this completely natural environment. Fortunately, we can study the patterns and courses of radioactivity because certain forms of it prevail for many years. Research at the atoll is a part of the Atomic Energy Commission program, planned and directed by men from the Applied Fisheries Laboratory at the University of Washington. Set up in 1943 to do research in radiobiology, the laboratory is headquarters of the Bikini Research. A staff headed by the laboratory's director, Dr. Lauren Donaldson, has been going back to the Pacific each year since the Crossroads tests. Dr. Donaldson, Alan Seymour, assistant director of the laboratory, and Dr. Arthur Welander, professor of fisheries, plan the research and field operations for the project, so that each visit extends a little further their knowledge of the radiobiological problem. Veterans of repeated trips to the Pacific, they have studied Bikini's islands and reefs and waters, studied the geology of Colorado or the ruins of ancient civilizations in America's Southwest. Months of planning reach a climax when the group moves to Kwajalein, big survey headquarters in the Marshalls, where members of the survey party and Navy representatives coordinate their operations for the overnight trip to Bikini Atoll, the last leg of the 5,000-mile journey. Equipment and supplies brought from the United States are checked and sent out to an LSI, especially equipped by the Navy as a floating laboratory for our field headquarters. We find Bikini little changed from year to year. Lines of coconut palms still mark the horizon. Even the old Crossroads Highway on Bikini Island is there, although partly overgrown now by vines and sprouting coconuts. The beaches are still white and lovely, but they carry the rusty tokens of the days the atomic explosion shook the lagoon. Here and there in the coral shallows, the rotting hull of an old vessel meets the breakers. Towers which once held automatic cameras still stand against the sky. The overall problem becomes clearer when you understand the atoll itself. Actually, Bikini is a coral saucer, 23 miles long and rising 1,500 feet from the floor of the ocean. The islands themselves, very low and frequently swept by waves during storms, rise only 5 to 8 feet above the water at their highest points. After the underwater explosion occurred, the saucer of Bikini retained quantities of radioactivity. Some settled back into the lagoon. Some of the fission products were blown or washed away. Many of these decayed at normal rates, and others were thoroughly diluted by the movement of sea currents, so that members of the group can safely swim in the lagoon and work on the islands. But there still is radioactivity in Bikini's life system, picked up in trace quantities by marine and land animals and plants, such as the algae or seaweeds. Part of the survey involves the study of the organs and tissues of fish that live in the coral shallows, and the organs of birds, like the fairy tern, who depend on insect and aquatic life for food. Radioactivity is still found in the bodies of the perennial beachcombers, in animals like the ghost crab. The radioactivity we find in the trunks and leaves of the trees and vegetation is drawn up through the roots with the water from the atoll's porous coral base. It's a strange and interesting fact that radioactivity continues and still can be detected, even in small amounts, in this place exposed to constant winds and a tropic sun, a place washed repeatedly by driving Pacific rainstorms. Our problem is to trace and study the nature of radioactivity in the life cycles throughout an aquatic environment in which an atomic explosion has occurred. The main station of our floating laboratory headquarters is 500 yards off Bikini Island, between the island and the 1946 target area. From this position, members of our group can conduct surveys and collections on Amen, Bikini, and Nu Island, and on the reefs and sand spits between. For a time, the LSI is moved into the target area itself to take samples of water at various depths and conduct dredging operations, hauling up materials from the bottom of the lagoon. Later, the laboratory ship is moved to the west end of the lagoon, where collections are repeated on Namu, Boro, and Erik Islands. To accomplish the necessary surveys and collections, the men and equipment are organized and ready by dawn each day. The landing party has to wait until after dawn to leave, because the approaches to many of the islands and areas of the lagoon selected for study are scattered with dangerous coral heads and reefs lying near the surface of the water. When the party gets to a point just off the coral heads, the men must transfer themselves and their equipment to a small rubber life raft for a long ride over the breakers to the beaches. A small camp is established, and then the teams scatter to their various jobs, some to work on land, some in the water. Monitors survey the islands with various types of radiation detection instruments, including the Geiger counter. Remnants of Crossroads equipment, such as this wreck on Nu Island, are monitored to keep check of the decay of radioactivity. If you think of a scientist as a man at home only in his laboratories, much of this work seems strange. Dr. Orland Biddulph, professor of botany at Washington State College, collects samples of vegetation, the coconut palm and the pandanus, plants which formerly provided food, clothing, and shelter for the Bikini natives. He strips the flowering top, certain tissues, and finally removes the flower of the plant. The coconuts themselves are given special study because of their importance in the native diet. Few varieties of animal life in the islands. Birds and rats are especially important because they are sensitive indicators of the presence of radioactivity. Frank Lohman, a research associate in genetics, traps rat specimens in simple but very effective devices made from tin cans and baited with food from the ship's galley. A major part of the fieldwork is conducted in the waters of Bikini, on the reefs and around the coral heads. Poison is spread by sifting it from cloth sacks, poison which forms a cloud in the water and stuns the fish within range. Then, with face masks and knives, we dive for fish specimens paralyzed by the poison. Sometimes the work must rapidly to gather specimens before the sharks move in. Few of nature's creatures are more colorful than fish taken from the sparkling blues and greens of the Pacific. There are the cornet fish, parrotfish, and pigfish, the surgeonfish with retractable spines of scalpel sharpness, and the Moorish idol, whose flat grace has made him a symbol of Pacific marine life. At the lower end of the scale of plant life at Bikini are the algae, colorful seaweed forms found in the waters or clinging to rocks, metal, or wood, and the plankton, tiny animal or plant organisms which are floating or weakly swimming in the waters. Both algae and plankton have been found to contain relatively high counts of radiation and are believed to play important parts in the concentration and movement of radiation in the lagoon. Dr. Kelshaw Bonham of the University of Washington and Spencer Tinker, curator of the Waikiki Aquarium of the University of Hawaii, collect hundreds of types of aquatic invertebrates. Invertebrates, or animals without backbones, are important specimens for study because usually they are not given to roaming far from their natural home, and any radiation absorbed by them will be held and stored until released in the normal term of life and death. Invertebrates such as this tiger cow ray laying eggs, hermit crabs, a proboscis worm, a spider snail, or even a sea slug may make noteworthy contributions to the radiation story because they hold radioactivity despite the physical changes of their aquatic environment. Samples of all the fish, invertebrates, rats, and birds are preserved in deep freeze lockers aboard the laboratory ship until they reach university laboratories. By discovering where traces of radiation exist in all these forms of life in the atoll, and by following it through life cycles, scientists are coming to a better understanding of the problem of radiation in or near water. Some specimens are captured in fish traps built of heavy wire mesh and baited with colorful objects, like a coffee cup from the ship's galley. From past experience, we know the strategic locations around the coral heads where the traps will be most effective, though traps are less dependable than poison spread around the reefs. Even the great coconut crab, while strictly a resident of solid ground, makes his contribution to research. This specimen, captured on Namu Island, is so powerful that he can shred a lead with his big pincers, a feat he is happy to perform. Because of the importance of plankton research, the laboratory ship is equipped with a big pump mounted on the stern of the LSI and capable of drawing up 10,000 gallons of water an hour from any specified depth of the lagoon, and equipped with valves for controlling water flow, and a set of nets of various gauges to trap the small planktonic forms as they are brought aboard in the salt water. Detailed notes are made of the time and place the samples are obtained, and depths from which the water is drawn. Because of our work in this remote shipboard laboratory with test tubes and infrared lamps, in the dissection rooms and the aquarium, gradually we are able to understand the radioactivity you and I encounter each minute we live. Natural radioactivity in the earth, the plants, even in the food we eat. Navy support by sea and air continued throughout the research operations, with an LST bringing fuel and fresh water. The field trip to Bikini is only the first phase of our work. After a voyage through the high seas playground of flying fish to Kwajalein and a long flight home, the final work, covering many months, is accomplished in the quiet of university facilities. Hundreds of samples gathered at the atoll are brought from cold lockers of the Applied Fisheries Laboratory to be dissected and prepared for examination by members of the Bikini team. All of the samples, whether fish, vegetation, algae, plankton, or invertebrates, are prepared for the final and most critical process in the laboratory, the ashing of specimens and quantitative counting of their individual radioactivity. At the atoll, Dr. Bonham gathered specimens of hundreds of crabs, snails, and other invertebrates on the coral reefs and in the surge pools. After this hermit crab gets its picture taken, it's weighed to establish its general size and development. Then a sample of its tissue is placed in an ashing plate and weighed so that radiation counts per gram of tissue can be determined later when it is sent to the laboratory to be processed. Another one of the identification steps, being completed here by a laboratory technician, is the identification of certain species of invertebrates such as sponges, which can be classified only by identification of their spicules, transparent bodies of bony material which support the tissue. In another part of the laboratory, work with the algae goes ahead. Ralph Palumbo, the algologist, who had to swim for his specimens at Bikini like nearly everybody else, processes a specimen of halimeda taken from the target area. Complete records are type, and the date and place it was. Everyone in the laboratory is preliminary work, which leads directly into the final studies of radioactivity. The ichthyologist has already done preliminary dissection aboard ship for already worked with a microscope to classify his specimens and make them ready for final tests. As liquid is, the plankton are poured into an ashing plate. Frank Lohman, who collected birds and rats on Bikini's islands, prepares the prop tissues for the next step in his research. Similar preparation has been accomplished with the samples of coconut and other vegetation brought back by Dr. Biddulph, who worked with the botanical collections. This is the beginning of the critical process. Each sample must first be weighed, and the original weight figures tabulated to establish the material, an important factor in later interpretation. Then, small pans containing the tissue samples are transferred to a hot plate under infrared lamps. When most of the moisture has been removed and the pans have cooled, they are transferred to a small muffle furnace. After 5 to 10 hours of baking, the specimen is reduced to ash. Water is added, and the mixture ground to an even consistency with a mortar and pestle. With a pipette, the mixture is placed on the small plate, a process that produces a uniform distribution on the plate. To assure the greatest possible accuracy in the detection of radioactivity, the drying must be repeated to spread the sample as a thin film on the surface of the plate. All of this preliminary work has been leading, in each case, to this, the actual counting of radiation. The plate containing the ash is placed in the small sliding tray of the nucleometer and inserted into the internal Geiger chamber for a predetermined period of time, where the sample will tell its story in radiation counts. Many of the samples brought back from Bikini tell their radiation stories in other ways, by giving us their radio autograph. This Bikini fish, a damsel, has been placed between small screens to keep the sample flat. When the tissues are thoroughly freed from moisture by exposure under the infrared lamp, the specimen is ready for the photographic process. When special film is exposed to its radioactive tissues, a radiation photograph or radio autograph is produced. In 1946, just after Operation Crossroads, this particular fish might have made his picture in a few minutes. But now it usually takes many days or weeks because radioactivity throughout the Bikini area has partially disappeared or has been scattered throughout many forms of life. After an ordinary photographic developing process, the result is the photograph on the right, made by the fish itself. The glow was produced by the exposure of the film to radiation contained by the fish tissues. Finally, all the material is gathered into reports for the Atomic Energy Commission. Annual installments in a continuing story, a history of Bikini radioactivity and its biological potential. The facts wait only for discovery and analysis.